Hi, good evening, everyone. I hope my voice is clear to all of you. I have shared a Mentimeter code on screen. For those who have just entered, please go to menti.com, enter the code, and you will be asked a question. Please respond to that question and submit your answer. And after that, there'll be a second question. If you haven't answered the second question, you can again enter the code and go to the question that I'm talking about. Please make sure that you're responding to two questions. I just wanted to take a inventory check on how the live classes are going on for you. So the first question is about how has the live classes helped you, while the second question is about if you think that these classes continue, what should your next topic of priority be from the Edmodo modules, which is the topic that you want me to reteach. So please go to Menti and respond to these questions and then we'll begin. I'm waiting for a couple more minutes because I think some more students are trying to join, and after that we'll start. Okay, I have actually started to receive responses. So you have written that lots of your doubts have been cleared. That's a good thing. Motivates you to study and pay more attention to biology. So classes have been helpful. Thank you. It has been helped me recheck my knowledge while the classes are going on. Okay. So you're supposed to recall a lot of information in short period of time. It's been helpful to clear doubts. Okay. Classes have helped me engage a little more with bio. Also be a little more participative than just completing modules and submitting them. Classes have a great way for me to revise previous modules and concepts. They have helped to clear some misconceptions. Okay, I'm glad that this has been helpful. I'm waiting for your responses for the second question and then we can begin. Enzymes, biomolecules. Okay, more discussion on cell ultrastructure, different functions of organelles like Golgi body. I think we have already covered these. So I'm looking forward to work on other subtopics. That's why I wanted you to tell me what is your next priority. More revision on microscopic structures. Okay, we can do some practice questions around it. Hmm. All right. uh, I hope, Gitanjali, you got disconnected. I hope you got a chance to answer the questions that I'm displaying on the screen. Were you able to go to Menti and share your responses?
Yeah, the I think ultra structure, the different functions. That is my response.
Okay. All right. Great. I will actually save all of this and compile it. And maybe in the one mail in which I'm sharing all the recorded classes soon, I will put up all these requests so that um, it will actually help me plan my next live classes. Hmm. So one reason why I'm not taking the topics in the way it is arranged in the module is because I am picking topics which students can do even if they haven't done the module. And I'm picking those topics which are more frequently asked in exam paper to start with. Gradually, we will try to see which pattern is fitting best for your learning in Avsara. Should we continue live classes like this, or should we do a mixture of live as well as Edmodo? The school is still deciding on that, and based on that we will see if we should continue the module sequence in our live classes also, or should we just focus on key areas where mostly everyone is struggling in the modules. Okay, thank you for your valuable information and tips. It's actually very helpful for me. Great. um, Yeah, so with this, I will now move on to our session where we will actually be continuing with the next set of organelles, that is the cilia, flagella, and microvilli. We'll be studying what is the key feature that differentiates each one from the other. After that, we'll do a quick review activity where you will check your understanding on everything that we have discussed about cell this by far, and a little detail about bacterial cell structure in terms of considering bacteria as a model of all prokaryotic organisms. What are some structures which are found in all prokaryotes, and what are some bacterial structure which are only found in specific type of bacteria? We'll see that. We'll also catch a glimpse of recalling what is the endosymbiotic theory and what is the evidence that this theory is may be true, and then close our discussion with features and characteristics of viruses. So the first slide on your screen is displaying an electron micrograph image of the microvilli. The singular of microvilli is microvillus, and mostly they are only found about 1 micrometer long. This is normally their length. Now, interestingly, they are only found in animal cells, and they are nothing but the cell's cytoplasm and the cell membrane, which has been pulled and tucked in the form of finger-like projections. So, do not think that microvilli are some things that are sticking out of the cell, but it is just the cell's own membrane which has become more projected or pulled out in the form of finger-like projections. So they are extensions of the cell. That means they will also have the cell membrane in their boundary. Where will you find microvilli mostly? You will find in, so your body has different kinds of tissues. Out of them, one type of tissues are the epithelial tissues. These are called epithelial. Epi means over. So epithelial tissues are mostly found in the covering of your internal organs, like in the inner lining of your respiratory system, inner lining of the reproductive system, inner lining of your digestive system. These are the places where you will find epithelial tissues, and this is the place where you will find epithelial tissue has been modified into these finger-like projections called microvilli. Now, why are they there? Why are they found on epithelial tissues? Because most of the time it is the epithelial tissues which have to do the job of absorbing something from the interior cavity of your body, and for that you need lots of surface. The more the surface, the faster and the greater amount of absorption will happen per unit time. So to increase the surface area, instead of having a flat cell membrane, the cell membrane is projected into finger like structures, and so that lots of cell membrane can fit in a small place. So because they're very densely packed and so small, you can see about 1 micrometer long, under the light microscope, whose resolution is only about 200 nanometers, you can only see the blurred edges of microvilli stack, a tuft of microvilli. That's why this image that you're seeing on the screen is not taken from light microscope, but it is taken from an electron micrograph. Uh, Under the light microscope, this will look very blurred. Only you can see the boundary. And under the light microscope, the microvilli looks like a hazy boundary of the cell, and that's why epithelial cells which have microvilli on them, they are called brush border epithelium, because the border appears as if the border of the cell is not smooth but has bristles or brush like structures. So it's this microvilli which gives that brush border appearance. Finally, the point in blue is always about the biological role. What is the function? So the function of this microvilli in your intestine, you already have studied. You have lots of villi and microvilli on the surface of these villi. So these increase the surface area of your intestine. In a small area of your abdomen, lot of surface of intestine can be accommodated if it has these finger-like projection, and along the length of these microvilli, food is absorbed from your intestine to the blood capillaries. Another place where the importance of microvilli is very significant, that is in your kidney. So in your kidney, when your blood gets filtered, all the toxic materials, excess salt and excess water gets uh, taken up by the kidney from the blood. But sometimes in the first round of filtration, necessary things also get filtered out from your blood, and those necessary things have to be brought in back into the blood. And for that, your kidney also has lots of microvilli, and when you will study A level, you will be studying about how kidneys microvilli help in reabsorbing minerals that should not get excreted out of your body. So I'll pause for 10 seconds so that you just quickly scan through the points once again, and then we can move to the next slide. You can also ask me if you have any questions here. Okay, are we good to go then? Or is there anything that you want to ask? Okay, I'm assuming there's no question. So let's see what's on the next slide. On the next slide, we have a outline diagram of a generalized animal cell, which shows the microvilli. So like I told you, microvilli is not something extra or additional appendage that is attached to the cell, but it is the cell's own cytoplasm and membrane, which has formed extensions or projections like fingers. So in this region, you can see a lot of cell membrane has been accommodated in just a small area. So imagine if the entire cell will have microvilli all over itself, and how much cell membrane can fit into just, how much surface area can fit into just one cell. More the surface area, more diffusion can happen, more materials can cross in and out of the cell. So absorption will be faster. Next, we are going to discuss about cilia and flagella. In our previous discussion of session 1, we had talked about centrioles and microtubules. Actually, cilia and flagella are made up of these microtubules. The only difference is these microtubules are not found in your cytoplasm, but they are extending out of the cell. And wherever, however long they extend, they get wrapped around by the cell membrane. So if you look at this image over here, it's showing one particular flagella which is wrapped around by the cell's plasma membrane. but this flagella has now extended out of the cell's cytoplasm and looks like a tail-like structure. And if it is a flagella, it will be one or two or three or four in number, few in number, and they are very long and they look like a whiplash. Okay. Whereas cilia are short hair-like structures extending from the cell to the outside, and they are present in tufts. They are present in large number, covering all over the surface of that cell. So a ciliated cell and a flagellated cell will slightly appear different. Um, Here you can see how, so basically both cilia and flagella are, internally they are nothing but bundles of microtubules, just like centriole. But centriole has no membrane around it, and centriole is found in the cytoplasm, whereas cilia and flagella are protruding out of the cell's cytoplasm or cell surface. Both of them are helpful in locomotion and movement. Now we have to understand that in case of prokaryotes, all prokaryotes are single celled. So if they possess a flagella, the flagella will actually help the prokaryote to move from one place to another. That's why we say that the flagella shows a whiplash like movement, like a whiplash. Whiplash are those long belt like structures which are used to lash the animal and scare them so that they carry the goods in the correct track. Like normally it's used on horses and all these animals which carry transport goods. So the flagella looks like that, those belts, and they move in this direction. As you see in this image, they move like a screw, corkscrew, whereas cilia are short hair-like structure and they show wave-like movement. Okay, so many cilia will beat together in a wave-like movement, and they will help in movement of material on the surface of the cells. So in our respiratory tract, for example, we have lots of cilia along our trachea and along our windpipe. Also in our nasal cavity, our cells possess lots of cilia or tiny microscopic hair-like structures, not the hair in your nose, but the microscopic hair-like structure on each cell of your nasal cavity. They are constantly beating so that mucus is always flowing along the air passages. And this mucus helps in trapping dust or any pathogen which is trying to invade your lungs. So it traps it and passes it into your throat so that your it goes into the stomach, and your stomach acids just destroy the microorganisms that are trying to enter your lungs. So that's one thing that cilia does. If the cilia is found in a unicellular eukaryote, means let's say paramecium, then all the cilia will beat rhythmically so that the paramecium can move from one place to another uh, as per its requirement. So that's why over here you can see in the last point I have mentioned that flagella and cilia, either they will help a whole organism to move, or if the flagella or the cilia are found in the tissues of any animal, then they are helping substances to move on the surface of the tissue. In case of animals, the most commonly found cell which has a flagella is the sperm in case of eukaryotes. But in case of prokaryotes, flagella are found in many types of bacteria. Uh, in case of eukaryotes, the eukaryotic cells are there who have flagella as well as cilia, but in prokaryotes, it's rarely seen that any prokaryote has cilia. Mostly prokaryotes have flagella with them for their locomotion. I have just mentioned a few examples here. Just take a few seconds to go through this slide, understand their structure and their function, and where they are mostly found. We have to just keep one thing in mind, that is, even though they are made up of microtubules, they are always covered by the cell's own cell membrane. So they should be considered as membrane bound. Is there any question that you want to ask about flagella or cilia or anything that you find confusing? You can ask by unmuting or in the chat box. All right. So then, shall we move ahead, girls? Are we good to go to move to the next slide, or do you have any questions? Okay, let's move ahead then. Here is a small tip which will help you not get confused in exams. Do not confuse microvilli with cilia and flagella. Most of the time, students confuse microvilli with cilia because they know flagella is one whip-like structure. So when they see many finger-like projection, they think those are cilia, but that's wrong. They are not cilia. Cilia will be there too. They will look like hair-like structures, but microvilli will look like finger-like projections. And also microvilli is a tiny projection where inside the microvilli you will not find any rod like structure, you will find cytoplasm, whereas in a section of a cilia you can see microtubules. Okay, because the cilia is actually a cylinder of microtubule covered by cell membrane. So be careful, look for cytoskeleton. If cytoskeleton is there in the projection, then it is cilia. If there is no cytoskeleton, that means no microfilaments are there. Those are villi, microvilli. A cilium is an organelle that is covered in cell surface membrane and contains 9 plus 2 microtubule arrangement. That means dimers of microtubule protofilaments join as 9 sticks together and form a cylinder. A flagellum is a large cilium, and cells have only one or few flagella, unlike ciliated cells. So, if you're confused between cilia and flagella, a cell will have numerous short hair like structures, those are cilia. But if it has two or three very long tail like structure, those are flagella. Next, I just wanted you all to take a quick look at the structure of the cell membrane. In Unit 4, we actually study the cell surface membrane even in more details. But in Unit 1, you need to know the basic um, characteristics of the cell membrane. That is, it is about 7 nanometer thick. It is the outermost boundary of the animal cell, but it is the second boundary for the plant cell, because a plant cell also has a cell wall. It is made up of a lipid bilayer, that is phospholipid bilayer, and in between the phospholipid bilayer there are proteins that are embedded. The function of the cell surface membrane is it makes sure that the external environment and the inside of the cell, that is the cytosol, has is partitioned properly, so that it creates a boundary for the cell, so that the internal environment and the external environment of the cell is separated. Therefore, the cell membrane guards molecules that enter and exit the cell. We say that the cell membrane is selectively permeable, not just semi-permeable, but it is selective about the type of molecule, whether the molecule trying to enter the cell is hydrophobic or hydrophilic. Based on that, the cell membrane looks at decides whether that particular particle will enter or leave the cell. Also, the cell membrane contains these structures called glycolipids and glycoproteins. Basically, you can see here the red color structures are proteins, and on the top, on the outer face of the protein, there are carbohydrate chains attached. So, carbohydrate is represented as glyco, and the rest of it is protein. So, we say this is a glycoprotein. These glycoproteins behave as receptors. They keep scanning and checking if there are any signals on the outside. Signals could be any chemical molecule sent by other cells. So these glycoproteins and glycolipids, they behave as the cell's own ID tags. They decide what should enter and what should not enter the cell, or what signal is coming from the neighboring cells. Also, these glycoproteins help in maintaining cell to cell communication, sending information to neighboring cells and receiving information from neighboring cells. Now, now that you have read about all these features about cell membrane, I have a question for you to think and share in the chat box. Can you view the cell membrane using a light microscope? If yes, why? If no, then why not? Think for a few seconds and please share what you think in the chat box. Please justify your answer. If you think no, then why not? And if you think yes, then why? Let's hear from everyone. I have only received a response from Pooja till now. Okay, Uzma says that it cannot be viewed. Uzma, your reasoning is not at all strong because electron microscope is necessary. That's why you cannot see the cell membrane in light microscope. That's not the reason. Gitanjali says, yes, we can view the cell membrane, but we can't see a very detailed image of it with phospholipids and other things. But you didn't give me the answer. Why? Why can we view, you think? And why can't we see the details? Give me that reason. If you're saying yes, then justify why you can see. If you're saying no, we can't see it under light microscope, then what is the reason? Okay, cell membrane can't be resolved by the light microscope. You have, Uzma, you are going on the right track, but you still didn't give me a complete reason. You are right that light microscope cannot resolve it, but give me data to prove it. You have data on your screen and you know about light microscope's resolution. Give those values to prove your answer. Others, everyone should share the response. Siley, what about you? Are you there or not? I'm not seeing your response in the chat box, Siley. Okay, Pooja says that the resolution of the cell membrane is 7 nanometer. That is, first of all, that is wrong. Cell membrane doesn't have resolution. Cell membrane is not a lens. Cell membrane is a structure. So yes, the thickness of the cell membrane is 7 nanometer. But the resolution of the light microscope is, the limit is 200 nanometer. That means objects which are smaller than 200 cannot be resolved, or they will not be distinctly visible under the light microscope. So yes, yes, you cannot view the cell membrane. So when we see an onion cell or a cheek cell under the microscope, we can still see the boundary, isn't it? But next time you see the onion or cheek cell under the microscope, you will notice you can see the boundary very hazily, very in a very blurred manner, but you cannot actually see the cell membrane as a dark line. You can never see that under the light microscope, it will look like a blurry line. The reason is our cell membrane is 7 nanometers in thickness, and 7 nanometers is way less than 200 nanometer. Our light microscope can only detect or show clear details of structures which are 200 nanometers and above. Make sense? So that's why our answer will be no. Cell membrane cannot be clearly seen under light microscope. You can only see blur edges. Okay. On your screen is a passage. Take a couple of minutes and yeah, Pooja, I understood. That's okay. So take a couple of minutes and try to fill in the words which are missing here. And then I will cold call and I'll ask you. If you have a pen and your notebook, quickly write down the missing words so that when I ask you, you are ready to share. Okay, your time starts now. I'll give you a minute and a half.
Is the dash between the cell
Cell surface membrane of a cell is a dash between the cell and the external environment. It is dash permeable. Extensions known as dash increase the surface area for dash or secretion. In cross
is dash
section,
permeable?
cilia have a dash microtubule arrangement. In animal cells, there is a total of dash centrioles. These are important in forming dash fibers during nuclear division. The vacuolar membrane is also known as the dash. Dash are cytoplasmic strands that pass from one plant cell to the next plant cell through cell walls. And the last sentence is the cellulose in plant cell walls provide strength and prevent the cell from bursting or dash. Just mention in the chat box that you're ready to share or you're done so that we can start discussing. So are we ready to share? Uzma, Gitanjali, Pooja, Saili, are you ready?
I required 1 more minute.
Okay. All right, are we done now? Pooja, are you done?
Yes.
Okay. And what about others? Uzma, Gitanjali, Saili? Okay. So let's begin. What have you written in the first blank, Gitanjali? Cell surface membrane of a cell is a dash between the cell and the external environment.
Boundary.
Okay, boundary or barrier, both will be correct. If you have written boundary or barrier, that's right. Uzma, it is dash permeable. What did you write for second blank?
Semi-permeable.
Semi or selective. Selective will be a more correct word. Semi-permeable was fine for 10th grade. For 11th grade, now we will practice to write selectively permeable. Okay.
Okay.
Next, extensions known as dash increase the surface area for dash or secretion. Pooja, you'll tell me 3 and 4.
Uh, 3 is microvilli
Correct?
and 4th is absorption.
Correct. In cross section, cilia have a dash microtubule arrangement. Who has written the answer for this? Uzma, have you written?
No, I didn't.
Saili, do you remember the microtubule pattern? We discussed this in the misconception slide. If you can,
Yeah, they are there are 9 patterns.
9
Three
plus 2. 9
There are 9
microtubules.
triplets.
Yes, so the answer is 9 plus 2 means there are 9 cylindrical rods, but each rod is a dimer. Means each rod is made up of 2 tubulin rows. Okay, so it's called a 9 plus 2 microtubule arrangement. That's correct. In animal cell, there is a total of dash number of centrioles. Who is going to tell this answer? Gitanjali,
Two.
how many centrioles are there?
2 centrioles.
2 centriole is the correct answer. Then next is there. These are important in forming dash fibers during nuclear division. Uzma, what is the name of those thread-like structures with which centrioles separate the chromosomes into 2 halves during cell division? If you remember, let's see if anyone remembers. Pooja, do you remember the name of the fibers?
Spindle fibers,
Spindle
right?
fiber is the correct answer. Spindle. Spindle fibers. The vacuolar membrane is also known as dash. Siley, what's the name of the vacuole membrane?
I'm not sure. Tonoplast.
Tonoplast is the right answer. What are the cytoplasmic strands that pass from one plant cell to another? Basically, these are cytoplasmic connection through cell wall. Gitanjali?
Plasmodesmata.
Yes. Plasmodesmata is the right answer. And finally, the last statement is cellulose in plant cell walls provide strength and either it prevents bursting or shrinking. So plant cells don't burst or shrink like animal cells because they have the cellulose cell wall, which maintains the fixed shape of the cell. All right. With that, our summary gets over. Let's move on. I hope most of you have got all your answers correct. If you've got all 10 on 10, then good job. If not, then with more practice, you'll be there. Don't worry. So now we'll do a quick review activity. In this review activity, I'm telling you what will come up so that you know. You will be asked some questions about prokaryotic characteristics. You will be asked some questions about endoplasmic reticulum, Golgi body. You will fill gap activities, and you will annotate a diagram of the bacterial cell. So I'm sharing this activity link with all of you in the chat box, and this is how your activity will look. Question number 1, you have to match the correct words together by pulling one word to the other. So whichever is the correct, you will just join them. Question number 2, you will fill these blanks. Question number 3 is interesting. You can see some words written in italic. So you have to just click on it and check whether that word is correct, it's fitting in the statement or not. If it is a wrong word, then you correct the word and write the correct answer over there. And in finally, question number 4, you have to identify these parts of the bacterial cell. Let me share it with you. So for this, I'll give you 6 minutes. It's 5:16 now. We'll come back at 5:22. You can start your work. Once you're done, submit it on my mail, on my Avsara mail, or you can send a screenshot on WhatsApp, whichever is easy for you. Please start working on it. I am here. So in case while solving the worksheet you face any difficulty, just unmute and ask. You have 1 more minute to quickly finish up whichever question you are at. Please let me know if you need more time. Okay, it is 5:22 now. Can you guys just type in or unmute and tell me how many of you are done, or if you need more time, how much time do you need? I guess it will be okay if you take a couple more minutes. Will you be done by 2 more minutes, everyone? 3 minutes, okay. All right. Okay, I'll wait for, let's get back at 5:25. Come on, be quick, girls. All right, let's quickly wrap up whatever we are writing and submit our work so that I can discuss the answers on screen. Please make sure that you share your final work or submit it to me either on my mail. I'm typing my email, Avsara mail ID in the chat box so that you can just copy paste it. I can check your work after the class. Pooja, what happened? Why did you not be able to complete it? Is there any technical issues or what happened? Okay, maybe you can all save this link and complete it and share it with me after the class. It'll barely take a couple of minutes. That's okay. You could, if it is not getting sent by submit, you can just send screenshots of your answers to me. That is also fine. So let's all come back and see whether our answers were right or not while we discuss over here. Please let me know that you're back on this call by telling in the chat box that I'm back. We are running out of time, girls. Please be a little quick. Okay, Saili and Uzma, Pooja, you are also here. I think Gitanjali got disconnected, so she has to join in. So on your screen right now, I'll just share the correct answers so that you also get a sense of how many answers you have answered correctly. The first question is connect the organelles with the correct information about them. Centrioles, centrosomes, and ribosomes. These are organelles which lack a cell membrane. Sorry, membrane. They are non-membrane bound organelles. So I'm going to match them with, where is the point? Lacking, organelles lacking a boundary. Smooth and rough endoplasmic reticulum, lysosomes, Golgi apparatus. They have single membrane. They are membrane bound, but they have a single membrane. So it's this one. Nucleus, mitochondria, and chloroplast have double membranes. They are organelles with double membranes. Cell surface membrane, it's partially permeable barrier, about 7 nanometers thick. Centrioles or centrosome, this is the region where spindle microtubules grow for cell division. So this is the correct matched pairs. How many of you got all of this correctly? Let me know in the chat box. Prokaryotic cells, let's look at question number 2, contain a group of organisms called bacteria. So all the prokaryotes mostly come under this group of animals, group of organisms called bacteria. Their DNA is dash, this is circular and not enclosed by a nuclear envelope or nuclear membrane, both are correct. They possess ribosomes and a cell wall made up of, Pooja, can you tell me what is the cell wall made up of in bacteria?
The word is very hard. I can't pronounce
So
it.
in exam you will write I can't pronounce. Will you say that in exam? Uzma, what is the word?
No.
Good. So this mark goes to me and no marks to all of you. That is peptidoglycan.
Peptidog.
What is the point? I told the answer first. The ribosomes found in them are of dash variety. Which variety? Ribosomes in bacteria. Which type of ribosome, Uzma? Can you tell? Okay, Uzma, I can't hear your answer, so I will write 70 as. All prokaryotes lack membrane-bound organelles. Some may have one or more whip-like structures. Just now we studied. Which is that whip-like structure that helps in locomotion? Flagellum or flagella. Okay. I hope all of you have written these words in this question number 2. Let's look at question number 3. Rough endoplasmic reticulum has 70S type of ribosome. Is it correct? Is it 70S type of ribosome on rough endoplasmic reticulum?
No.
Remember, rough endoplasmic reticulum is only found in eukaryotes and in the cytoplasm, not inside the mitochondria. So this will not never be 70S. This will be 80S ribosome. Okay. And where will this ribosome be? On the inner or outer surface of the endoplasmic reticulum? On the outer surface. This is correct. Doesn't need any correction. And for what? For the synthesis of protein. Okay. Now they are saying within the dash of rough endoplasmic reticulum, proteins can be transported. So should this be wall or should this be lumen? Are the proteins
Lumen.
synthesized in the wall or in the lumen or in the cavity space inside the ER? It is the
Lumen.
yes, lumen, so that proteins can be transported to other areas. Now comes smooth endoplasmic reticulum function. It helps to synthesize carbohydrates. Is it carbohydrates? Should we be okay with this answer?
It's lipids.
It's lipids. Okay. SCR has a smooth endoplasmic reticulum has a more tubular appearance. Is this correct or wrong? It is actually correct. Smooth endoplasmic reticulum looks like a tube. So it will be tubular only, tubular appearance. But rough endoplasmic reticulum, we saw they are like flat sacs, right? And these flattened sacs have a name. Are those flattened sacs of rough endoplasmic reticulum called thylakoids? Gitanjali, do you think this is thylakoids?
No.
Then what is it?
It's a something.
Something, right?
Last time also you
Something
told me.
exam.
It's cyst something.
Right? Cyst something in exam.
No.
Your examiner will take a bucket of popcorn and enjoy your answers and laugh. Okay. Cyst something. So the answer is cisternae. Transport vesicles move protein to the chloroplast. So this transport vesicle is coming from endoplasmic reticulum. Will it move the protein to chloroplast? Siley? Where will the protein go?
Golgi body.
Yes, correct.
Golgi body.
Golgi body or Golgi bodies, whatever, where they can be destroyed. Golgi body destroys the protein.
No.
Then,
It will be modified.
yes, modifies and packages them into Golgi vesicle, modified and packaged into Golgi vesicles. Powerful digestive enzymes called acid hydrolysis. This acid hydrolysis is correct or wrong? It's actually correct. Hydrolytic enzymes are acid hydrolysis. Other name for hydrolytic enzyme is acid hydrolysis. And where will they get packaged into? Which organelle? Centrioles. Is centrioles correct? So this is our correct word. And who is going to tell me? Pooja? So these hydrolytic enzymes, which are actually the protein made or packaged in the Golgi, where will they be put into? Into which organelle? Centrioles.
Lysosomes.
Yes. Lysosomes. These function to do lysosomes use hydrolytic enzymes to join materials like bacteria or break down.
Break down.
So a better word is digest. To digest materials like bacteria or old and damaged DNA. What does lysosome do?
Organelles.
Apart
Like
from digesting foreign pathogens entering the cell, what else does it digest? Think, recall, video you watched.
old and damaged organelles.
Correct. So that's our paragraph. Okay, now we'll annotate this diagram. You are going to tell me, okay? Unmute and tell, so it will be faster. What is this structure, Gitanjali? First box, whip-like, lash-like structure.
Flagella.
Flagella. Are you sad, Gitanjali, about the fact that it is flagella?
No, no.
Okay, you sound sad. Uzma, what is this thread-like thing in the bacterial cell? What should I write? Tell fast. Can't hear, Uzma. You all can type in the chat box, but I'm not being able to see. Siley, what is this? This thread-like structure?
DNA, circular DNA.
Okay, you can write DNA.
DNA.
You can write genetic material. Okay, you can also write chromosome. Everything is correct. Okay, third box. Look at the arrow. What is it pointing? And what should you write over here? Pooja? Any idea, Pooja? Yes, correct. This is the cytoplasm. Then the next one, these tiny dots in the bacterial cell. Uzma, this time can you tell what are these dot-like structures? You can't hear, only noise I could hear. Your voice is not audible. You can type in the chat box. Okay, this is actually your 70S ribosomes. Let's look at these tiny hair-like structures coming out of the outer wall of the bacteria. Gitanjali, what are these called if you remember from the module? Gitanjali?
Microvilli.
No.
No.
Microvilli are
Pili.
only found in animal cells. Is bacteria an animal? It's a prokaryote.
Yeah. Is
So
it
this,
pili?
yes, this is a pili, okay, or pili, whatever. The function of pili we'll discuss after this annotation. What is this small circular structure? Siley, can you tell? Pooja, Uzma, Gitanjali, what is this tiny ring-like structure?
Circular DNA.
Circular DNA, it has a name. It's called plasmid or small circular DNA. Correct. Next, this line. What is this line indicating? Which part of the bacterial cell? Who can tell? Anyone? Unmute and tell.
Capsule.
No. Watch carefully.
Cell wall.
Capsule is always
Cell wall. Sorry,
outermost.
sorry, sorry.
Cell membrane.
Cell
Yes.
wall.
Cell membrane is the right answer. Then what is this green color border? It's just outside the cell membrane. So what should it be?
Plasma membrane.
Are cell membrane is plasma membrane. Again, one more plasma membrane. This is a boundary outside the cell membrane. So what should it be? Cell
Cell
wall.
wall.
What is the cell wall made up of? Difficult word. Pooja, tell me. Don't tell. In exam you will forget. Peptidoglycan. Okay. Peptidoglycan cell wall. Last. What is this outermost covering? Some bacteria have it, some don't have it. What is it called? Gitanjali?
Capsid.
Capsid is found
Capsule.
in virus. This is bacteria.
Capsule.
Capsule, or this capsule is actually very slimy. So it's sometimes just called slime layer. Okay. So this is the structure. These are all the parts. Okay. Let's quickly take a look at all the parts and more details, because after this our time will be up. I want you to take a look at all the parts, and in this diagram the functions of these structures are also mentioned. Okay, so flagellum helps in movement or locomotion of this bacteria. Nuclear material is where all the protein codes are stored in this bacteria. So whatever characteristics the bacteria is showing is found in the genes inside the DNA or this nuclear genetic material. Cytoplasmic is a jelly-like matrix in which all the organelles and ribosomes are floating. So they are the main soluble fluid inside the cell where all the enzymes and everything, soluble materials are found. 70S ribosomes, they help in protein synthesis of this bacteria. Plasmids are small circular DNA molecules apart from the main DNA. Always remember, you are going to study about plasmids in detail when you study antibiotics. So bacteria have this extra DNA, and this extra DNA contains genes which protect bacteria from all the antibiotics that we make to kill them. So actually plasmids have proven to be the most important tool for biotechnologists to study genetic engineering. So they use these genes in these extra DNA that the bacteria possess, and these genes contain different protein codes that actually protect the bacteria and make it more and more disease resistant. So all the disease resistant power or capacity is because of these plasmids. The more plasmids a bacteria has, the more powerful it is and more difficult it is to kill that bacteria. Okay, so the plasmid is a very important, small but very important piece of DNA. Pili, these are hair-like projections when the bacteria is sexually exchanging its genetic material. So they contact, one bacteria will contact its neighboring bacterial cell through the pili. Not only for reproductive purpose, but also for cell-to-cell communication, they use this pili. Pili is also used to sense changes in their environment. Temperature, pH change, all of that is sensed by these hair-like pili. Cell wall is peptidoglycan. You know, the function is to protect the cell from any external entry. It is made up of not cellulose but peptidoglycan. And the final outside slimy capsule layer, some bacteria have it, some don't. The function is the same, that is, it protects the bacteria from getting digested. If the bacteria is a parasite and it is living inside the intestine of a host, there's a chance that the cell will get digested, but the slime layer protects that from happening. Okay, so this is a very general idea of mostly structures that are found in bacteria. In the next diagram, you actually seeing an electron micrograph of an E. coli. Colonies of E. coli live in our large intestine symbiotically. That means without harming us, they help us in maintaining a healthy large intestine environment. But they're opportunistic. They can cause infection if we have any sort of immune response related diseases, then they become pathogenic. Now, if we want to study the structure of the E. coli has some characteristics which are common with all bacteria and some are very specific to E. coli. Look at this diagram on the left hand side. This is one bacterial cell whose structure has been divided into two parts. One side is showing some extra features that some bacteria have, and the right hand side is showing general features that all prokaryotes have. We just discussed the general features, that is ribosome, circular DNA, cytoplasm, cell membrane, cell wall. Some extra features that you will find in some bacteria and some prokaryotes, not all prokaryotes, that is the flagellum, capsule I told you, or not all bacteria have the capsule. This is additional protective layer. Now, some bacteria can do their own photosynthesis and they can also produce their own ATP and they do the cellular respiration, but we know that bacteria cannot have membrane bound organelles, right? So they're very well evolved. How do they do that? How do they manage? They The cell membrane will infold and form many folds. With many folds there will be more surface area, and on this surface area there will be enzymes and proteins which are actually helping in making ATP. So we say that in the eukaryotic cell, the job that our mitochondria, chloroplast, endoplasmic reticulum do in specialized bacteria, because they don't have membrane bound organelle, they have these infoldings. And those functions and those work or operations happen in these infoldings. And these infoldings actually have a name. These are called mesosomes. I'll see if I can write it over here. So these structures over here, these are called mesosomes. And these mesosomes correspond to the function of our chloroplast, plant cell chloroplasts and animal cell mitochondria, as well as other membrane bound organelles. Then we were talking about plasmids. These are small circular DNA. Lots of them may be present. Each of these DNA contain genes which make the bacteria resistant to some kind of antibiotic that we try to make and attack the bacteria. The pili are for attachment. So if one bacteria has to attach with another bacteria to communicate some information, they use these pili. And these are some extra additional features. And these are the general common features that you see in all bacteria. Okay. All right. Next, I want to quickly conclude. I will actually not start the endosymbiotic uh, theory today because we have run out of time. So rather I would conclude our discussion here. I'm just trying to erase this thing. Okay. Yes. So in our next discussion, I want to discuss about endosymbiotic theory a little more, and about viruses. Once we do that, we are done with this unit, and I will take into account what you shared in the discussion in the opening questionnaire, where I asked you what are some topics that you want to learn about. So I will see if we can plan some lessons around biomolecules and enzymes. But in the next week, a day in advance, I'll let you know when we are having a common discussion Zoom time, where we will talk about the endosymbiotic theory and the viruses. If you have any further questions, please feel free to ask me in the WhatsApp group or directly message me on WhatsApp. And that's all for today. Thank you very much for participating.